with us. Thanks for being here. Please also remember the evaluation forms on your way out, just so that we can have proof to the GME office that we are indeed having grand rounds. Again, thanks for being here. I'm Jeremiah Hopkins, the Chief Resident, and I would like to give a warm welcome today to our speaker, Dr. Greg Ordway, who will be talking with us about blueberries and antidepressants. Dr. Ordway received his BS in pharmacy and a Ph.D. in pharmacology from The Ohio State University College of Pharmacy. He also is a neuropsychopharmacology trainee and postdoctoral fellow through the University of Psychiatry at the University of Pennsylvania School of Medicine. Prior to coming to ETSU, he worked in several roles, including at Case Western Reserve University and the University of Mississippi Medical Center, including Associate Professor, Director of the Division of Neurobiology and Behavior Research, and Assistant Vice Chancellor for Research. Dr. Ordway came to ETSU in 2005 as the chair and professor of the Department of Pharmacology. He is also a professor in the Department of Psychiatry and Behavioral Sciences, and also a former interim chair and professor of the Department of Biomedical Sciences. He is currently a senior advisor to the Dean for Research. Among a number of other awards he has received the Distinguished Faculty Award for Distinguished Achievement in Research here at ETSU. He has also received the Jack L. Bill Post-Baccalaureate Award from the Ohio State University of Pharmacy in 2012. He's on a number of committees here at ETSU, including the Grand Rounds Committee for the Department of Psychiatry. He's authored and co-authored dozens of publications, multiple chapters in a book entitled Brain Norepinephrine, Neurobiology and Therapeutics. Dr. Ordway, thanks so much for being with us today.
I have no control over the volume of it, so can you hear me now? I can raise it up and make it a little louder if you'd like me to. I'm really excited there's a lot of students here today, and generally, I think normally Grand Rounds is kind of rather formal and people don't ask questions during presentation. I urge you, if you want to, you can raise your hand and I'll answer your question during the presentation. We can keep it as informal as we possibly can. Today I'm going to be talking about some of the research that my laboratory has been doing in depression and suicide, but we also are studying autism, and maybe I'll kick myself, but I'll come back next year and tell you about our research in autism, which we're also very excited about. So here's my detailed list of financial disclosures and conflicts of interest. So if that bores you, I can tell you that I am beholden to the National Institutes of Health and American Foundation for Suicide Prevention, and ETSU, who is also helping in funding my research. I'm also beholden to my collaborators, Craig Stockmeyer, who's been a major source of brain tissues for our research, and then my local collaborators, the doctors Zebedee and Zebedee, and Tim Perry, who's a medical student here at ETSU, Michelle Chanley, and Jessica Crawford. Point out that what I'm presenting to you today is really some ideas that Attila had in my lab, and after a lot of arguments and discussions, he convinced me to move the direction of the laboratory in this area that he had some bright ideas about, and that's what I'm going to present today. It turned out to be quite interesting. So I thought I'd start with just as a prelude, a little history of antidepressant drug development. Is that not loud enough? It's an analog method of adjusting the volume. The history of drug development. So, and the history here and the lesson is that we have not been very successful in this realm. So, pharmacological treatment of depression started in the mid-1900s or early 1900s. Amphetamine, wow, this is really fast. So some things work well, huh? started with amphetamine, and then actually the major advancements in development of drugs in depression were two. One was, I guess you might say amphetamine is, in a sense, this is a drug that clearly raises or elevates mood, but it's got some problems. And those problems were eliminated with the development of imipramine. The problem with amphetamine was that it has robust effects on dopamine and therefore has a lot of abuse liability. Whereas imipramine was a drug that targeted the monoamines, serotonin, and norepinephrine without touching dopamine, and this turned out to be a very effective antidepressant. Well, as effective as they get, even today. And then the next major development, even though multiple drugs are being developed, all these drugs are manipulating norepinephrine and serotonin. The next major development was not really a paradigm shift at all. All it was a development of a drug that did not have the toxicities that the older drugs did. So fluoxetine represented it. It's considered a major advance in the field, not because it's some magically better drug or anything. It's just, well, it is, but just because it's less toxic, that's all. Not because it's more efficacious. And so throughout the course of history in the development of pharmacological therapy for depression, there really has not been a major move in the field since imipramine, which is a very efficacious antidepressant drug. Some recent studies, relatively recent studies, suggest that it may be the most efficacy you can get out of any antidepressant drug, with other drugs like it, but less toxic, also having this rather unique property of inhibiting norepinephrine and serotonin uptake both. So the bottom line here is that over 60 years of drug development, and we really haven't stumbled across a drug, we stumbled across imipramine. That was an accident. We haven't developed a drug that has significantly better efficacy than the drugs that were discovered by accident. Of course, if you go back to amphetamine as the beginning, you'd say, well, that was a major development. But of course, if you remember back, those of us who were around, I probably wasn't studying antidepressants back then. I was very young. But this is an advertisement from Smith, Kline, and French from a magazine article. And back then, Smith, Kline, and French was telling us that dexedrine, which is Spancel, it's an extended release amphetamine, was the basic antidepressant drug. And of course, we know better than that now, but this is an example of how you need to listen to the drug reps very carefully with a certain amount of scrutiny. Luckily, Smith, Kline, and French left a little room for the possibility that amphetamine wasn't the basic antidepressant drug, and we now have better drugs. But, as I said, every drug that's available today for depression are drugs that are manipulating as a primary target norepinephrine and serotonin. So that's where we are, and unfortunately, antidepressant response rates in patients is relatively low, about 50 to 60% response in depressed patients. But that response is measured in terms of a greater than 50% reduction in symptoms. It doesn't mean they're back to normal. Remission occurs in far less than 50% patients who have depression and take antidepressant drugs. Approximately 30% of depressed patients will not remit after multiple trials of drugs. And I think that the statistics are that you have about a 30 to 35% chance of getting better with the first antidepressant drug you take. So it's pretty low odds. So if you calculate this in terms of remission rates, being there's 30% who never remit, and do the math with the number of people who are clinically depressed in the United States, you find out there's about 2.4 million people who are essentially untreatable or only marginally treatable, which is, a travesty. And that is despite a number that I calculated. I went to the NIH website and tried to figure out how many $1 billion have we spent on depression research and antidepressant drug development. And you can't really get that because they don't have statistics that go way back. But in the past 6 years, we spent $2.5 billion for NIH research. And I estimate that the NIH, over the course of its history, has funded about maybe up to $15 billion of research on depression and antidepressant drug development. Then add into that tens of billions of dollars that drug companies have invested in, although they're much more conservative and they're just tinkering with existing molecules. So we have this situation where we have a class of drugs, all very similar in their action, that are not very good at what we want them to do. Yes? Sure, yeah.
This is looking at pharmacology alone and doesn't
Yes.
ECT or TMS.
No. ECT is a little bit better.
Yeah, I'm thinking we have a little better rate
Yeah.
on that when we do ECT.
So, yeah, right, you're right. And once you fail all this, then you go to ECT, which knocks you up, and maybe you'll have 10 to 20% that don't respond to ECT, maybe. Right. Thank you for pointing that out. So why, I think what we have to conclude is that we are not at the point of treating mental illness like we are in the treatment of physical illnesses. Much better at treating physical illnesses than we are mental illnesses. And why is that the case? Why do we not have good antidepressant drugs, or better ones than what were discovered by accident back in the 40s? And I found this I found on my Facebook. I posted this cartoon I thought was apropos to today's discussion. And this is, if physical illnesses were treated like mental illnesses, then this is the kind of suggestions that people would make to somebody who had a physical illness. At the toilet, regurgitating from the flu, and they say, have you tried, not having the flu? Or a guy who's cut his hand off, the advice from a friend is you just need to change your frame of mind, then you'll feel better. Or the guy who's had a heart attack lying in bed, and the suggestion is, well, lying in bed obviously isn't helping you. You need to try something else. So one thing that I think that has stifled drug development and research in depression is the stigma associated with mental illness. So the funding for the research is lower than physical illnesses. Although there tends to be there's a trend and change in that thinking, but that's been the history, and we're stuck with that history in the present day. The other thing is that research in the area has been, up until just a few years ago, heavily influenced by dogma in the field. And I've been in this field trying to get funding, and back before, I'd say 2005, if you were not studying norepinephrine or serotonin in your research on depression, then you would have to come up, and you're required to have a big, long justification of how what you are studying relates to norepinephrine and serotonin. So that mentality was entrenched at NIH, at NIMH. And what's happened in the past 5 years is complete flip-flop. So now if you are studying serotonin or norepinephrine, you got a low likelihood of getting funded. That's just the way it is now. We've completely flip-flopped. So you're unlikely to get, you can look at the rate of grants on serotonin, they've dropped off precipitously. And I had one of the few grants on norepinephrine up until a couple of years ago, and they decided I wasn't going to even resubmit it because I know the mentality and the shifting of minds at NIH. So despite that all the drugs that we have available to treat depression are noradrenergic and serotonergic, or both, the research in the field moved on, searching desperately for something else, and that's what I'm going to show you today is some work from my laboratory. We're looking at something pretty far afield from that. The one thing that is very clear from the literature is that stress is a major initiator of depression. And this is very well accepted and very well documented in the literature. This is a paper by Kindler. I'm sorry. These buttons are so close together. If I would have designed this, I would have put that button further apart. But I didn't design it. This is a paper by Kindler in 1999 showing the odds ratio for onset of major depression in situations of different life events which are stressors. So clearly, this is just one of many papers that have demonstrated that stress is a major risk factor for the development of depression. If you take home one message of the day, you'll note here marital problems that raise your odds up 14 times. You'll go home tonight and take your significant other out to dinner and a movie. And that is, I will consider a success of my seminar. So a few years ago, Susan Epple and Elizabeth Blackburn reported these data showing that high stress levels shorten telomeres. I'm going to tell you what a telomere is in a little bit. I think you mostly probably know a little bit about telomeres. But what she looked at were two groups of subjects that were caregivers of a debilitated relative, some debilitative illness. And she classed these as those taking care of relatives who were really ill or severely debilitated versus those that had more mild caregiving roles. And they were age matched. And she found that the telomeres in bloods collected from these individuals were shorter than the ones that were in a lower stress situation. And she also showed that the chronicity of the caregiving, in this case, you could see this as a chronicity of the stress, was significantly correlated with the telomere length negatively. So the longer you undergo this stressor, the shorter your blood telomeres get. Now, this is a kind of a messy relationship. Obviously, there are a lot of other factors involved, but this was statistically significant. So what is a telomere? A telomere is a non-coding repeat DNA that's at the end of the chromosome, and it caps the chromosome. It's actually a complex of proteins and DNA. They cap the chromosome and they protect it from degradation. If you don't have it there, then your DNA will degrade and the cell will die. And there is an enzyme, telomerase, actually several, involved in maintaining this length. In cancer, telomerase is very often constitutively active, maintaining length so that the cell can divide and divide and divide and divide forever and stay alive. But in normal cells, where it's less active, each time a cell divides, the telomere is shortened because of this lagging single-strand piece. There's something called lagging strand synthesis. You don't have to know that. Just know that when the cell divides, it gets shorter. And with age and with continued cell divisions, this is why we don't all get cancer, the telomere reaches a critical short length that stimulates pathways in the cell for apoptosis, and the cell goes into senescence and then dies. So this is just a schematic diagram of telomeres showing factors that are involved in telomere shortening and protection of telomere length. So as cells age and through replication and division, the telomere keeps getting shorter and shorter until it reaches a critical length, stimulates p53 and other apoptosis factors in the cell that results in senescence and apoptosis. This process is then, so telomere shortening is perceived then as a sort of a proxy of cellular aging. The shorter your telomeres, the more aging that's going on. And replication isn't the only thing that drives this, and what we're going to be focusing today is on oxidation, oxidative damage to DNA. Oxidative damage to DNA over the lifetime also shortens the telomere because telomeres are very sensitive to oxidation, and that oxidation causes clipping away of the telomere. And then there are enzymes called DNA checkpoint enzymes that try to repair that damage, and there's elongation by telomerase as well. So in 2006, Naomi Simon at Harvard looked at, based on the studies by Epple and psychological stress and telomere shortening, she looked at telomeres and leukocytes from major mood disorders patients and observed a significant reduction in major depressive disorder, telomere lengths in sites from major depressive subjects. She also saw this in bipolar disorders with anxiety. And this was the first paper to show telomere shortening in blood cells from major depressive subjects, and this has been replicated quite a number of times now. Now, I will point out that telomere shortening is observed in a lot of illnesses, cardiovascular disease, diabetes, Alzheimer's disease, so it's not specific for depression, but it's found in depression, and people have repeatedly demonstrated in various studies. In fact, this is a paper that was just published this year in Molecular Psychiatry. It's a very high-profile journal. And this group looked at over 1,000 major depressed subjects, collecting leukocytes and then measuring telomeres. And they not only observed a significant reduction in telomeres in depressed subjects, but a dose-dependent and chronicity effect. So the more severe the major depression, the shorter the telomeres in the blood cells. And the longer, I'm not showing it here, the longer they're depressed, the shorter the telomeres in blood cells. What's kind of interesting about this is that they have patients on different lengths of time on antidepressants, and the antidepressants do not seem to modify, are not a modifier of this effect. This is illustrated a little better in this paper. This is another paper by Hartman, that another group, in a smaller group of patients, but these are major depressives, and they show a significant reduction in telomere length in leukocytes. That's not modified by lower doses of antidepressants or higher doses of antidepressants or ECT. So current pharmacological treatments do not seem to modify the telomere shortening that's going on in major depression. So in the field, historically, if one saw this data, and I would say when they published this data, people think, well, it's not important because antidepressants aren't correcting it. So it's real common in the literature now for people who study depression pathology is they discover some pathology and then they go to some animal model or look in patients and say, look, we found this pathology and look, our antidepressants fix that. So therefore, it's very important to depression. In fact, that's what they use as the argument that it's important to depression because antidepressants fix it. But antidepressants aren't very good at doing what we want them to do. So I see this fact that antidepressants don't modify telomere shortening as an opportunity. This is something that our current drugs aren't doing, and maybe they should be doing. And maybe this is at least one reason why they're not very effective. And we haven't been able to come up with better ones. So Adela got this idea that we should study this. And we spent a lot of time talking about it before we actually did anything. One thing that was kind of a downer was people had looked at telomeres in the brain. So you've got this telomere shortening in blood cells. It's very reproducible. But, you know, my lab is sort of brain-centric. We tend to think that the brain might have something to do with depression, not blood. So blood can be a proxy maybe of what's going on in the brain, but you have to look in the brain to see if that's going on in the brain to see whether that's really, you know, any bearing on the illness. So there were two studies at the time when we were talking about this that had been published already. One was this French group, and they looked at occipital cortical tissues in major depression in control subjects. These are postmortem brain tissues. And they saw no difference in telomere lengths. And I'm showing you the means and the standard errors. So this is extremely tight data. No change. And then there's another group that looked in cerebellum and saw the exact same things. No differences in tissue, in the brain. And after those two studies were published, people said, well, what's going on in the blood is a proxy for something that may be relevant to what's going on to the brain, but the same thing's not going on in the brain. Well, I'll argue that, rightly so, that these are studies of brain tissues that contain a lot of different cell types. There's reason to believe that not all cells are going to be susceptible to things that might shorten the telomere, oxidative stress being one of them, a very likely contributor to shortening of telomeres. So, for example, in the hippocampus, there's two regions, CA1 and CA3. Pyramidal cells in those two regions very close to each other. The CA1s are exquisitely sensitive to oxidative stress and die. And in the same animal, right nearby, those neurons are spared. We don't know why. There's definitely a difference in their susceptibility to oxidative stress. Likewise, in white matter, astrocytes and oligodendrocytes are in white matter, and astrocytes are resistant to oxidative stress, whereas oligodendrocytes are very vulnerable. A lot of things going on in an oligodendrocyte that makes them vulnerable to oxidative stress. So he reasoned that the blood cells studies that have looked at telomeres in depression and other illnesses are looking at a very well-defined population of cells. They're not all the same, but they're pretty closely similar. Whereas the two studies that were published looking at brain tissue, these are very different tissues. They grind up a chunk of tissue. There's probably maybe 200 to 300 different types of cells in that chunk of tissue. So you would look at, what they're measuring is a population average. And if it's in a small population or a population that's small in their sample, they would never pick anything up. So we set out on a course to look at telomere links in major depressives using postmortem tissue and looking at specific cell types. And we got our brain tissues, as I mentioned, from Craig Stockmeyer, as we always do, most always do. And we looked at about 12 major depressive subjects. These are major depression at the time of death. This is using a brain bank that's funded by the NIH for the past 20 years. And these are verified major depressives at the time of death, and they didn't have antidepressants. They weren't taking antidepressants. Not that that's probably a factor in this case, but whenever we do these studies, we try to keep antidepressants out, at least in the initial studies. We matched them as best as we could for smoking, some alcohol exposures. Most of the suicides died of, most of the major depressives died as a result of suicide. Most of the controls had heart disease as a prominent cause. And remember, heart disease is a contributor to telomere shortening too. And then we used a method called laser capture microdissection, and it's not that important that you know the details of the methods, but I thought I'd show you this because 10 years ago this would have been science fiction, but now we use this routinely. I'll see if I can get this to work. Might not be able to. I'm almost afraid to hit anything here. Oh, here we go. So this is that's a pyramidal neuron that's being circled on the microscope. It's being cut out by a laser and then captured onto a little disc. So we have this instrument here at ETSU, and we laser captured specific cell types using this technology, and then we can study those specific cell types. That's a tissue section cut from a human brain. It's been stained, in this case with crescent violet, but you can use any kind of stain you want to identify cells. So we looked at two brain areas. We looked at the uncinate fasciculus and BA10 white matter. The uncinate, this isn't, we just didn't randomly do this. This bundle of axons passing through the uncinate fasciculus are coming from prefrontal cortical regions and projecting down to the amygdala. So this is they're carrying emotional information. That's why we went there. Information related to are being passed through this bundle of white matter, bundles of axons in this white matter. So we looked there, and we also looked in prefrontal cortex white matter. Because age affects telomere length, we were very careful to match the ages of our subjects. And I'll show you some of these data. Again, it's not that critical that you understand the details of the experiments and so forth, but more important that you get the concept here. So we looked at astrocytes first. We had a lot of data in our laboratory showing astrocyte pathology and depression. That's sort of how we got into looking at white matter and astrocytes and oligodendrocytes. But we thought maybe that some of the damage that's been reported by us and others in astrocytes might be a result of telomere shortening in these cells. Oh, gosh. But we labeled astrocytes with a marker and then plucked them out with a laser capture microscope and then isolated DNA and measured the telomere lengths. No difference in major depressives compared to control subjects in either white matter region. But when we went to oligodendrocytes, back to the same subjects and captured the oligodendrocytes, we found a significant reduction, a robust reduction in telomere lengths in oligodendrocytes from uncinate fasciculus and BA10 white matter both in major depressives compared to control. Here we're using a marker to identify these with a laser capture. So this was the first, we've published this already this year. This is the first time that anybody's looked at single cell types in the brain at the issue of telomere shortening, and we were able to identify telomere shortening in a group of cells that it's already known to be susceptible to oxidative stress damage. So this started the ball rolling, basically, that finding. We right away we got real interested in the telomere shortening and this idea of accelerated aging that has been proposed to occur in major depression based on the blood studies. And we knew that telomere length is maintained by this enzyme telomerase, that it's shortened by oxidation of nucleotides because the telomere is, as I said before, exquisitely sensitive to free radicals. And we knew that there is a defense system in your cells that's made up of several enzymes that protect the cells from free radicals. And we knew that there are DNA damage checkpoint enzymes that correct oxidation-induced DNA damage. So we wanted to take a look at these various factors, and I can show you some of these data that we've generated so far. First of all, it's not really known. It's known that psychological stress increases oxidative stress and induces oxidative damage in the brain. This has been shown in humans and also been shown in rodents. But how that happens, nobody really knows. I'm not going to be able to answer that today, but it is a really critical question. And it's been proposed by various authors that it's this state of elevated oxidative stress and free radicals is induced through a stress-induced increase in cortisol and insulin that results in elevated cytokines, et cetera, et cetera. There's a lot of hand-waving in this diagram and not a lot known. But for now, we don't know, but we do know that stress does increase oxidative load, and the brain is exquisitely sensitive. I mean, there's a high oxidative load in the brain because it's very active, unless you're sleeping right now, in which case you should wake up and get oxidative stress going. I'm not talking to you, Mary. So oxidative stress is what we wanted to focus on, and we wanted to get an idea whether there might be changes in oxidative stress defense in oligodendrocytes from major depressives compared to controls. So oxidative stress results in the liberation of a number of free radicals. One of the major ones is the superoxide anion. And when that's generated in a cell, there's a host of enzymes that make up oxidative stress defense. And three of these are, actually there's two superoxide dismutase, which dismutate the free radical to hydrogen peroxide, which is further detoxified by catalase or glutathione peroxidase. So these were genes that we could easily look at in our, well, not that easy, but we could study in our oligodendrocytes captured from major depressives. And we looked at these in both oligodendrocytes and astrocytes from major depressives and controls. And we found significantly lower levels of all four antioxidant enzyme genes in major depressives compared to controls. So the upshot here is that there's lower expression of the genes, for whatever reason, we don't know why, that would normally, these genes protect you from free radicals. In oligodendrocytes, but in the astrocytes where we didn't see telomere shortening, we saw no differences in the levels of those genes. In many cases, there were lower levels of those genes in the astrocytes, in the oligodendrocytes compared to the astrocytes. So our data here suggest if these gene expressions translate to the activity of those enzymes, that there's less activity on the stress defense side, which you would predict would result in more free radical damage to these cells. That is the prediction based on our initial data. So we've now been looking for evidence that there is free radical damage in oligodendrocytes. And Kate and Adela generated these data looking at lipid oxidation. There are kits for these things. It's amazing what you can buy on the internet. There are kits for studying lipid oxidation. And here we're just taking white matter. So far I've just shown you all that data are from oligodendrocytes and astrocytes captured from white matter. And here we're just collecting the white matter because these methods cannot be applied to the tiny amounts of tissue we get from laser capture. So white matter studies, and we measured lipid oxidation in the same major depressives, some of them. We're not complete. This is preliminary data. Major depressives in control subjects. And we found that there was elevated lipid oxidation even in this small sample size of about 4 to 6, somewhere in there. Elevated indices of lipid oxidation and elevated oxidation of DNA. DNA we were looking at is 8-hydroxy deoxyguanine. Guanine is the nucleotide that's exquisitely sensitive to oxidation, so that's why we looked at that. That's the one that there's a lot of in telomeres. So it does look like, at least in the white matter from the same areas where we collected the oligodendrocytes, that there is elevated levels of oxidative damage. It doesn't tell us whether the oxidative damage resulted in the telomere shortening. So Adela developed this method. It was a published method, but he applied it in a unique way to look at telomere-based, we're going to look at this side first, telomere-based lesions. Now, these could be oxidations, and there are other lesions that can happen. We don't really know whether it's oxidation, but the data would highly suggest that there is increased oxidation of the telomere. So he amplified just the telomeric DNA and looked at the levels of what we would refer to as base lesions. And he did observe a significant elevation of base lesions in the telomeric regions of DNA isolated from oligodendrocytes from the white matter. This is an enzyme. This is one of those checkpoint enzymes that I talked about. He also looked at PARP1 expression in oligodendrocytes for major depressives and controls. You see it's highly elevated. This enzyme is like a little detector. It goes along and finds the DNA damage and then fixes it. So an upregulation of the enzyme we would predict is occurring because there's elevated oxidation going on. So the cell's trying to respond to this by upregulating a checkpoint enzyme to fix it. We don't have to talk about this, but we also measured telomerase, and it was low in major depressives in oligodendrocytes, but not in astrocytes. So this is the enzyme that is not very active in brain cells, but its expression was lower in major depressives compared to controls, suggesting there's less capacity to lengthen the telomere. So let me just summarize that data, and then we can talk about the implications of it. So what our data has shown is that shorter telomere lengths in these susceptible cells, oligodendrocytes, and it's characterized by reduced levels of telomerase, reduced oxidative stress defense enzyme expression, and an elevation of DNA damage checkpoint enzyme PARP1. And on the other side, we have evidence of oxidation of nucleotides, so that, and in fact, in telomere nucleotides, evidence of telomere lesion, DNA lesions on telomeric DNA. So our data really we started out really interested in telomere length, but what we've sort of shifted to is that we realize now that telomere length studies, all the people that are doing this in the blood, what they're really measuring, it's a proxy, we believe it's a proxy of oxidative stress and oxidative damage to DNA. That's what's interesting. Not so much the telomere shortening, it's just it's just one of many indices of telomere of oxidative stress. So we predict that based on this schematic here, that because you have lower expression of these enzymes and psychological stress being a part of major depression and major depression itself being a stressor, that you have a state of elevated free radicals that are attacking DNA. And we've looked at oxidation of DNA, and we know it's elevated, and we know there are more lesions in the telomeres where there are lots of Gs, and this is a very sensitive region to oxidative stress. But so the real story here is not so much in telomeres, but telomeres as a proxy of oxidative damage to susceptible cells. And these free radicals attack all components of the cell and would be expected to cause all kinds of different damage and dysfunction to a cell. So what is an oligodendrocyte? What does it do? I mean, it's not a neuron, so it's not important, right? Well, oligodendrocytes have a lot of functions. There's actually a lot of different kinds of oligodendrocytes. It would be nice if there were just a few cell types in the brain, but there's multiples. And the one that we studied using a marker CNP is a mature myelinating oligodendrocytes. So these cells set in the white matter and some are in the gray matter, and they send a little projection out to the axon. Here's the neuron sending its axon somewhere, and they myelinate the axon in layers. And this is in segments. And these segments are interspersed with the node of Ranvier. And the nodes of Ranvier are what make transmission of electrical signals along the axon fast or slow. If you don't have any myelin and no nodes, it's slower transmission. If you have a lot of nodes, the transmission jumps and it speeds up a lot. So think about a computer or a circuit board. The speed that information flows has a lot to do with how it operates and how it gets processed. So it turns out that years ago it was thought this was just a static thing. Your cells are your neurons are myelinated, and then you go on with life. Now, this is a very dynamic process, and this is just one more level that your brain manipulates and modifies transmission of information from one place to another. Some axons have no myelin in the brain. Some have segments of myelin. Some have a segment myelinated, then a blank space, and then another segment myelinated. And it turns out that this is constantly changing, and it's modified by a variety of things, drugs and environment. So we would predict that, based on our studies, that these cells, which are inherently sensitive to oxidative stress, and I didn't go into the molecular and cellular details of that, but some of those reasons are very well understood now, why they're very sensitive. That our data suggests that these are exquisitely sensitive, and in major depression, in a state of elevated superoxide anions, that there is damage to these cells that then results in probably different levels of myelination or demyelination that's going to interfere with transmission flow. And in critical regions like the uncinate fasciculus that's carrying emotional information that's being processed at nanosecond speeds, that those tiny little changes are going to result in dysfunction and abnormal transmission of information. So is there any evidence in humans that there's white matter pathology other than this data that we have? There's actually a ton of data out there. So white matter hyperintensities measured using diffusion tensor imaging is a way of measuring the way water molecules, basically is what they're measuring, the movement of water molecules through a white matter. There are, in this review that looked at a total of 19 studies measuring white matter hyperintensities, 14 of the 19 found elevated white matter hyperintensities in major depression compared to control subjects. White matter hyperintensities is not really known what the cellular basis of those are, but they're little bright spots on the image. And major depression is not the only illness, again, that demonstrates white matter hyperintensities. But it's one, and it's been reproduced many times. So we would speculate that those white matter hyperintensities are a result of dysfunction or decay of oligodendrocytes in those white matter regions, possibly secondary to oxidative stress and a unique susceptibility of those cells to oxidative stress. So this is just a summary of those data that I've just talked about. How are we doing on time? Let me just move on. I've just summarized that for you. I don't think I need to go through it again. So I just want to tell you a little bit about the theory that we're developing. So psychological stress is, as I said, is a major factor that raises your risk robustly for the development of major depression. And it's been known for many years that psychological stress manipulates those old pathways that we've always talked about in terms of the biology of the illness. Particularly, it depletes norepinephrine and serotonin. So I'm not saying in any way that everything that's been done so far, and I'm a person who's studied the norepinephrine part for most of my career, that stuff's not important and not going on. It is going on, but there are other things going on, and one of those things that's contributing to the illness is likely to be white matter pathology. So what we're after is trying to understand what is going on in white matter. So I'm developing this flywheel brake model of white matter pathology. So the outer wheel here is spinning, and it's being driven by oxidative stress damage, or damage mediated by oxidative stress. And again, I'm focusing on white matter here, recognizing that there's a lot of other things going on in the illness. So psychological stress elevates free radicals, and that accelerates aging in susceptible cells. And the susceptible cells that we've studied so far are the oligodendrocytes in white matter. This results in some dysfunction. This disrupts communication between brain regions, disrupts behavior, and will reduce adaptive and coping behaviors to psychological stress. So this wheel's turning, and as it turns, eventually, if it's not slowed down, you end up with a mood disorder through the contributions of effects in white matter. Inside the wheel works like a clutch, like a flywheel or a brake, and it might serve as a flywheel that if you don't have good coping skills, you have some gene polymorphisms that lower the expression of some of your antioxidant enzymes, making matters worse. You're exposed to some drugs, or you've had previous insults. Then it works as a flywheel to speed up that outer wheel and push you into a mood disorder quicker. It may work as a brake. If you have really good genes, you've got great coping skills, you're in a good environment, maybe taking antidepressant drugs would help, although the data would suggest not. Then it might break this process and keep you from ultimately developing a mood disorder. When's he going to talk about blueberries? So what the data shows so far is that antidepressants don't have any effect on this process. So that just based on the telomere aging alone, now that's not all the other effects that are going on, but on the telomere shortening, antidepressants do not seem to modify telomere shortening, current antidepressant drugs. There are a lot of data over the years that have studied vitamins and antioxidants and depression. It's a highly controversial area. But, you know, it's very difficult to do those studies. There's not really any really good studies out there on. There's some fairly decent studies on vitamin E and its effectiveness in depression. But they're rare. And they're not case controlled. They're not. But if you think about it, why should that be so difficult to understand? People do clinical trials with fluoxetine and can't demonstrate significant antidepressant effect because there's such a robust placebo effect in these studies. So imagine trying to modify somebody's diet, which you can't even, it's very hard to control. But I think that the data here would suggest that we need to look at this much more carefully. And I've been doing this with a collaborator. We've gone to an animal model, and Sharon Campbell in the old biochemistry department is really interested in, she's always been interested in vitamin E and has been studying it in cancer. And we're now trying to look at some of these things in animal stress model and looking to see whether vitamin E does anything and can protect the brain. I'll just throw this out there. We use a social defeat stress model. I'm not going to go into great detail, but it's a very psychologically stressing environment for a rodent. And when you put them in, put animals in this, it's basically a bully mouse with a bully mouse, or a bully rat, they get anhedonic. So they normally consume a lot of sucrose, sucrose-flavored water, but when you stress them heavily, they quit drinking the sucrose, and that's considered an index of anhedonia. And then what she did was Sharon treated them with a mixture of tocotrienols, which is an isoform of vitamin E, and it reversed the stress-induced anhedonia. This has actually been demonstrated previously, but she was doing it to confirm it. And what's interesting about it, it reversed effects on adrenal weight, which is a measure of chronic stress. And then she was able to show that nitric oxide, which is one of these reactive oxygen species that's generated during oxidative stress, that the tocotrienols reverse that elevation of this free radical. And just preliminary data demonstrating that the stress induces, this is a measure of oxidative stress in the amygdala. There's a massive increase in oxidative damage in the amygdala following the stress. So we think that this will be a good model for us to go on and look at white matter and oligodendrocytes, et cetera, and try to parallel our human studies in an animal situation where we can manipulate it and see whether we can find some unique compounds that might be useful for preventing oxidative damage to white matter. Also look at what might be the behavioral outcomes of white matter damage. I think that's my last slide. So I'm going to open it up to any further questions you might have. Eat blueberries. I eat them every day. I'm a believer.
Seems to me that the general idea is chronic exposure to glucocorticoids that probably specifically affects the frontal area of the brain. And as I'm listening, it strikes me that chronic exposure to glucocorticoids may very well deficiency of why we are great.
Yeah, right.
Okay. Further insight into that.
We don't have any data on that, but we're real interested in that.
What's the hand wave?
Right, exactly. We don't know the mechanisms, but we don't. We've started to think about this. You know, first of all, we don't know whether the white matter is affected in all regions equally. I have a grant application in right now with NIH trying to convince them to fund us on this. We want to look at some other regions because it's expensive to do the work. See whether it's just everywhere or is it in areas where there's, you know, ocean processing going on. And then the question is, it might be cortisol, elevated cortisol. So we thought, well, we can look at, you know, glucocorticoid receptors on oligodendrocytes, and maybe they have more of them, and that would make them more susceptible to the effects of cortisol. Or maybe they have the same amount, and they're just more susceptible that when cortisol goes up, it drives down the expression of those enzymes that are normally protecting you. We don't know that either. It could very well be. There's some data to suggest that that steroids can downregulate oxidative stress defense. But it's a mixed, some data suggests it upregulates it, may be very, it's hard to know right now. We really need those kinds of studies you're talking about.
Questions. The first one was, you know, you mentioned about 50% response with, or remission, I guess, with antidepressants. Now, the STAR-D trials would tend to suggest after 4 levels of trials, it might be closer to maybe 2/3.
Yeah.
But again, that's with
That's right.
combinations and whatever. So that perhaps is a little bit better.
Yeah, a
that
little
doesn't
better.
include the ECT that Dr. Hendricks
It
mentioned.
did
So
include the ECT?
it did not.
Oh, it did not.
Not so, but it did look at MAOIs and combinations of medicine. So
Still leaves about 30% of people.
agreed, totally agreed, and I agree. And I think maybe ECT picks up on that little percent, whatever, you know,
Yeah.
whatever. So that's one piece. The other piece of that is, I wanted to ask you about is, you know, you talked about how antidepressants may not be having, well, clearly aren't having an influence on telomere length. What about the implication? I mean, is there anything in terms of what psychotherapy might do as far as that, because that's working completely, well, we don't know exactly. I mean, it may all
Right.
go into biology again and whatever, but
Yeah.
we don't know. And then the other thing is, you know, there's a lot of discussions about resilience these days about, you know, why people exposed to certain amounts of stress come out. And that certainly can be genetics and environmental. But do you, like, you'll often see people with this, let's say, abuse or whatever, you know, and you'll find that some people come out of it without major depression, and some do. Do you feel like most of that really is genetics, or do you think that there may be some protection early on beyond genetics that would make a difference?
Well, your first question about behavioral therapy, I think that's people are working on that, and I wouldn't be surprised that it does slow down telomere shortening, but I don't think the data's out yet. because the drugs don't do it, but they're working on specific systems, whereas behavioral therapy is working on the whole system, right? So I wouldn't be surprised if that were better. And then the second thing about, well, I'm sorry, what? Resilience is, yeah, what constitutes resilience is probably multifactorial. Learning, development, genetics, environment, so many things play into resilience that, who knows? That's right. Absolutely. Yes, they're all not psychiatrically normal. All the controls have been screened through first degree next of kin, and they are all psychiatrically normal. So, and most of them are heart disease. So the way I interpret that is, based on other studies, heart disease is associated with shortened telomeres. So when you have heart disease, it's not as robust effect. So depression's worse. It's causing an accelerated aging relative to heart disease, which is also accelerating their aging. I'm sorry, Tom, did you have a question?
Well, I don't know if it's a question, it's more of a musing.
Okay.
I've come across in the last several years a handful of cases in which individuals with pretty advanced hemochromatosis that was not medically appreciated by their psychiatrists were being treated for mood disorder, and in some instances didn't get better until the metabolic problem was recognized. I read up on it because I was curious about it, and I recall that iron was identified as a source of oxidative stress. So I'm just wondering, in the context of what you're talking about here, how would you separate oxidative stress from psychological sources in the pathway you've described from any of the other pathways that might take you to the same place?
Well,
does it matter? Is that a population that needs to get studied and so forth?
Well, I think inflammation is definitely a source of free radicals. And inflammation is pretty common, and there's a lot of people that think that it's underlying major depression. So it's hard to separate those things out. At the level that we study it, there are a lot of factors. And for example, people are taking NSAIDs. They're taking anti-inflammatory drugs, and we didn't keep track of who's taking anti-inflammatory drugs and who didn't. And we know that inflammation is a contributor to free radicals. So we are not able to separate those things out. That's why we need to go to rodents and do this reverse translation where we can parcel out those things and try to figure out what it is that really is the major driver of the white matter damage, if in fact that does occur. And then we can look at different things that might be driving that. And I'm very interested in the concept of polypharmacy. I grew up I'm a pharmacist by training, and we of all the the negatives of polypharmacy. It's bad, polypharmacy. Too many drugs, bad. But there's reason in my mind to think that what we should be doing in depression research is focusing on, rather than trying to find treat people with ketamine. And I don't know if you know that recent literature, but where you're just about, it's like taking a sledgehammer and hitting somebody and making it better. We ought to be thinking about taking our existing drugs, which are, reasonably good, 60% of people maybe get better, or 60 or 70, and then adding on something that's hitting some of the systems that those antidepressant drugs do not. That's what I'm really interested in, is more of an add-on adjunctive therapy to existing drugs until somebody makes a major discovery. Because I think what we're studying is, not all cells are susceptible, so not everything's going to be affected, and it may be just certain systems that are affected that are contributors. They may not be the causal in nature, but they're contributing to this dysfunction. So,
Dr. Ordway, that's a very nice presentation that you gave today. I have one question in regards to the telomeres. When we have antidepressants that are on board patients, what do you think is actually happening to the telomere lengths after the 8 to 10 weeks in which an antidepressant kicks in. And by the way, as a plug for my grand rounds, I'm going to be talking about ketamine on December 5th.
Oh, I'm sorry, I didn't mean to disparage
So-
ketamine.
So we'll get in, we'll look at that.
Well, the data suggests that antidepressants aren't modifying telomere length. But antidepressant drugs, there's a lot of studies showing antidepressants do have antioxidant activity. But I tell you, those studies, they're using high doses and concentrations of antidepressant drugs to get that. So some are a little more antioxidant than others. I don't think that's a factor. But the current data suggests that antidepressants do not modify the course of telomere shortening.
But is this only in that short time frame, or because
Yeah.
over a longer period of time, have they looked at that, whether or not there is an effect?
So in the big 1,000 patient study, I don't think I looked at that section. I tried to find more details about the antidepressant part because I had this similar kind of question. Okay, how long were they taking the antidepressants? And they didn't report that. So I just not known right now. You would think if antidepressants are working also as an anti-anxiety drug, that they would be lowering the stress in the individual and that should modify the course. But so far, nobody's been able to show that. The data to date indicate it doesn't modify. It's not a modifier.
Mechanisms of disease progression, you're not worried about decreases in BDNF
Yes.
on brain volume.
Right.
And accompanied by those decreases or increases in oxidative stress. Are these independent factors, or is there some relation between them, some common mechanism?
I would think they're independent factors. The reduction in BDNF is mediated through the catecholamines and serotonin. So you deplete those through stress, and norepinephrine, for example, stimulates BDNF production. So when you deplete it, you get less BDNF. I don't think that's probably related to the oxidative stress pathways. I think they're probably independent. Now, there may be some overlap in it here and there, but mechanistically, those seem to be different to me.
What do you know about no-go A and agents that are coming down the pipeline for treating depressants, multiple sclerosis, may be being used for treating demyelinating diseases, and oligodendrocytes that is seen with depression.
What was a compound?
No-go A?
No-go A?
No-go A antibodies.
I don't know anything about it. So sorry. I'm relatively new to the oligodendrocyte field. Just we wrote our first paper, and I don't know about the use of drugs in demyelinating diseases and whether they have any efficacy. Don't know. It's an interesting idea, though. Carl?
It's been mentioned there's several pathways to oxidative stress, and one of the ones that I thought would be important to mention as a common one clinically would be vascular complement. So I was wondering to what extent you thought that vascular compromise is a source of oxidative stress in addition to chronic psychological stress is important.
What-
Looking at the etiology.
Right. In our sample, we did not have, I think we ruled out vascular problems in the major depressives. Is that not right? None of those, they're all suicides and they did not have cardiovascular disease. Is that right, Kate? Yeah.
Yeah, however,
So-
this can occur at the level of the microvasculature.
Yes, and then difficult to detect. Right.
Another,
And we know that our major depressive suicides, Kate tells me this, but we haven't done anything with the data, that the microvessels they have a space outside of them that looks like cardiovascular disease. In the major depressives without cardiovascular disease. So it may be playing a role.
another comment I had, it used to be thought that neurogenesis was related to the development of neurons and astrocytes. Now it's also recognized that oligos
Yeah, that's right.
included, and that with chronic stress you get an actual increase relative to oligos that form. And I wondered to what extent you would comment on that.
Did you review my grant? I mean, because there's a criticism we had to deal with, is that Oligodendrocytes are not a static population of cells. You're always making new ones, just like neurons and astrocytes. So it's conceivable that a change in the maturity, a population of mature versus younger oligodendrocytes, because of stress or depression, could change telomere lengths. And we are specifically going to answer that question and get funded. But it's a really good question. I don't think so. I don't think there's enough oligodendrogenesis going on that could shift the population that much to cause a change in relative telomere length, but it's possible, and we have to rule that out. So. Not that many, probably about, I mix fruits, so probably about 40 blueberries a day or something like that, 30 or 40 blueberries a day. I feel good. That's why, right? That's right. That's right. Oh, thank you very much.